Why can a microscope reveal a whole hidden world, while your eyes can't even see a single bacterium? What's the real limit, your vision, or the light itself? Here's the punchline: microscopes don't just zoom in. They capture tiny details by controlling light, how it spreads, how it bends, and how it enters the lens. Your eyes are amazing, but they're built for survival at human scale, not for resolving features smaller than a few tenths of a micron. When we say invisible, we usually mean too small. But the real issue is resolution, the ability to separate two nearby points as two distinct things. You can magnify a blurry photo 1,000 times and it's still blurry. That's exactly what happens if you magnify without improving resolution. Light isn't just a straight line ray, it's a wave. And waves spread. When light passes through a small opening or around an edge, it diffracts, creating a soft pattern instead of a perfect point. That wave spreading is the enemy of tiny detail because it smears sharp features into fuzzy blobs. Even an ideal lens turns a point into a tiny disk of light. Put 2 points too close together and those disks overlap, your image can't separate them anymore. That's the diffraction limit. For visible light, the practical best resolution of a standard optical microscope is roughly a couple hundred nanometers, around 0.2 micrometers. So what makes microscopes powerful? Not the 1000x label. It's how much light the lens can collect, and from what angles. That's called numerical aperture. Bigger aperture and better optics capture more of the scattered light that carries fine detail. More detail captured means sharper resolution. Sometimes microscopes even use oil between the slide and the lens. Why? Because oil bends light more like glass does, letting the lens capture wider angles of light without losing it to refraction. That boosts numerical aperture, and suddenly tiny structures that were borderline become crisp. Another problem: many microscopic things are nearly transparent. Your eye relies heavily on contrast, and clear cells don't give much. Microscopes add contrast using tricks: phase contrast, darkfield, polarization, staining, methods that convert subtle differences in how light passes through a sample into visible brightness changes. Fluorescence microscopy goes further, tags specific molecules with dyes that absorb one color and emit another. Now the microscope isn't trying to see the whole cell, only the parts you marked. It's like turning on tiny lanterns inside the sample, so the structure you care about pops out of the darkness. But what if you want to see smaller than visible light allows? You switch the probe. Electron microscopes use electrons, which have much shorter wavelengths than visible light. Shorter wavelength means the diffraction limit shrinks dramatically, so you can resolve incredibly tiny features, down toward the nanometer-scale and beyond, depending on the method. Your eyes aren't weak. They're optimized for a wide field of view, fast motion, and decent resolution at arm's length, perfect for real life. Microscopes are optimized for one job: forcing waves of light, or electrons, into a controlled system that preserves tiny detail and boosts contrast. That's why they reveal the invisible. Now I'll throw the next why at you. Why do some microscopes need vacuum chambers to work at all?